Hello, everyone. Great to be with you again. This is Doug Beall, interventional radiologist in Oklahoma City. And earlier in the meeting, we've had the data presented for the early feasibility studies for HYDRAFIL, which is a hydrogel within the disc. This is a not new product that is currently being tested and has been granted breakthrough status by the FDA this past year. So I'd like to talk with you a little bit. It's an interesting product that's a long time in the making. I'd like to discuss first how this came to be. So this is a mixture of PVA and PVP, polyvinyl alcohol, polyvinyl pyrrolidone, and this was initially started back and developed back in 2004. And this is designed to be a nucleus replacement to augment the disc. And this went in initially as a solid, then it was heated to form a 2 millimeter long tubular structure that looks a lot like spaghetti. And this was designed through a larger perforation, and the annulus designed to be a nucleus replacement. And this was the size of the replacement was determined by a balloon, and the nucleotomy was made prior to putting this in. And one of the downsides about this is, although the clinical results initially were optimal and the product worked well, it tended to have the tendency to expulse after placing it in situ when certain investigators were trying to place this. Certain investigators had good results, the other ones didn't, and whenever they didn't have good results, specifically, the implant tended to work its way out. And the first formulation, pragmatically, that was implanted was called Jellyfax, and this was Jellyfax was a tubular implant. Exactly the same implant as I previously described. This was developed by Dr. Tony Lohman out of Drexel. And whenever this was placed and placed well, it had great clinical results and did well for an extended period of time. And so once it was discovered that it intended to pop out, a redesign and reformulation phase was entered where the implant was designed to develop a material that was placed as a liquid injected through a smaller gauge needle, and this was started in 2008. So the idea was to inject the material as a liquid, and then it would form itself into one solid mass. In fact, the Jellyfax, when it was put in, if you put it under stress and strain and place it in a machine with, for example, 10 million cycles or even far less, it comes out as a solid, and it forms and links together by hydrogen bonds. So as long as it stays in an aqueous environment, such as like it's in the body, it can break apart and actually heal itself. It's kind of an amazing polymer. So the idea was to inject it as a more of a flowable, that way getting it through a smaller gauge needle without the need for the big nucleotomy, to be able to inject it and to have it mold and meld together over time. And this is incredibly resistant, very tenacious material, as this can undergo 10 million cycles, and now it looks like it looked like it had been stressed and strained at all. And so starting in 2008, this was the goal, to develop an injectable. So what was added to the PVA PVP was polyethylene glycol in small amounts. It was found that it didn't change the substantive properties of the gel, but it did allow it, when heated, to be placed into a cadaver, an animal, by way of a needle. And this is an example. It was placed through a 19 gauge needle and put into the cadaver, and it filled neatly the nucleus pulposus. And so with the addition of polyethylene glycol, this was made into a flowable. So this was designed to be a simple procedure, a simple injection into the disc, the same as discography, or the same as any type of nucleus augmentation with stem cells. It had optimal biomechanical properties, as we'll see here in a few minutes, and it mitigates the risk as it fills the fissures and the portions of the annulus that are injured, and the portion of the annulus that tears during disc degeneration. It will grab and meld itself into a solid and thereby stay in place. So the Jellyfax material was heated to 95 degrees C to soften it up enough to get it in through the nucleotomy. This material, with the addition of the polyethylene glycol, is heated up to 65 degrees C. This becomes a liquid that's easily flowable with a good viscosity in terms of Pascal seconds. And this is able to be injected into the disc. And when it hardens, it forms the typical hydrogel that gave rise to the good clinical results that we're seeing initially. So as mentioned, the injectability, it's a semi viscous material. If you measure the viscosity, it's between 30 and 185 Pascal seconds, of course, depending on how hot or cool it is. The thermal dose to the tissue is acceptable under 55 degrees, and it does form a continuous implant, and the swelling is mild. It swells a little bit more after it's placed, but not significantly. And it will not, importantly, protrude through a 5 millimeter hole, which is far bigger than the 16 or 17 gauge needle that is designed to inject it through the annulus into the nucleus pulposus. The modulus of Young is optimal, and it is impregnated with sterile barium, thereby making it very visible under fluoro. So one of the comparisons in Europe is gel sticks, and gel sticks had the similar problem with expulsion, but it did have an optimal clinical profile. And the re-engineering of the hydrogel, the new product instead of Jellyfix, is called HYDRAFIL, which is injected through a 16 or 17 gauge needle, hopefully solves this problem. And this is an example of pain and functional improvement associated with gel sticks. And this is a European study. You see fairly profound improvements in pain and function from the initial placement of the gel sticks all the way out to 6 months. But again, this had the propensity to expulse, propensity to pop out of the annulotomy site. The new HYDRAFIL material can be injected through a 16 or 17 gauge needle. There's no adverse effects noted, especially in the early feasibility studies. This is designed to show augmentation of the disc. The disc height is maintained as long as the hydration is maintained, which of course it is within the disc. And as I mentioned, the mixture of the polymers don't substantively change the optimal biomechanical nature of this. The material is warmed to 65 degrees and it liquefies and then can be injected. It becomes a solid at body temperature. So this is kind of the polar opposite of a cement. As a cement heats up, it becomes more viscous. As this cools off, it becomes more viscous. It's easy to see under fluoro. And the pre-procedure discogram, and this is important, predicts the fill pattern of this. So I find it very beneficial to do a procedure discogram to see where all the cracks, crevices, and annular tearing are in the disc, so you know exactly how much and where to fill. So initial studies, so no migration in animal studies, all the way out to a year, and there's actually animals far longer than that out now. And it does, whenever you inject it as a liquid, it does adhere to the surrounding nucleus pulposus and annulus fibrosus, and it can be used as a surrogate. Not only does this is space occupying, that holds the disc open, it's kind of like what I perceive as fix-a-flat for the disc. Every time we inject into the disc and pop it up, we wish it could stay there. And this time the material's semi-solid can actually do that, especially after it hardens. And unlike a lot of other treatments, it works best in discs that are very degenerated. And so as I mentioned, we have early feasibility studies. This was the design of the early feasibility studies done by our friend Dr. Alfonso Spath, who presented the EFS data at this meeting for the very first time worldwide. It was designed to have 20 subjects, but was expanded to 50 with a primary endpoint of safety, followed by pain and functional improvements. And to date, all 20 patients are completed. We have another site coming on board, in addition to the initial site in Barranquilla, Colombia, coming on board in Cali, Colombia, and this is to be initiated very soon. So let's take a look at those results. These were presented previously, but I want to just take a look at the pain and functional improvement results to show you give you an idea about the magnitude of improvement. So back and leg pain, this is typical of patients with degenerative disc disease. They have problems with not only low back pain, but with sciatica. Leg pain tends to be easier to treat in general than back pain. Back pain tends to be one of the most refractory things that we have to treat. And I'll call your attention to the fact that the back pain improvement was a whopping 78%. In fact, not only was it good, it was better than the leg pain improvement at 3 months. In addition, the function, which is fairly typical of lumbar spine, is function is as good or slightly better than the pain improvements. And this was the case here with an 85% improvement in oswestry disability. This can go to something that's on the average severely disabled to something that's not disabled at all. So these results are quite optimal. And this is ongoing, as I mentioned, rolling this up to 50 studies. The FDA granted breakthrough status for ReGelTec, who makes HYDRAFIL, on December 4th of this past year, which is a very big deal. The pilot study is due to start in the United States in the summer, hopefully in July. And we've already started screening patients and already have more than a dozen patients identified as candidates for this. And so hopefully we'll be implanting HYDRAFIL this summer. So in summary, this is a long run for a short slide. This is a long time coming, primarily because of the initial formulation had the tendency to expulse out of the disc. And something I hadn't mentioned before, the technology was purchased. So Synthes was developed. This was developed with the contract with Synthes. They were bought 2009 by J&J, and this technology was shelved for a while, bought back by the inventors with reemergence and re-engineering of the material. And the new formulations show as good a clinical results with a greater degree of injectability through the new reformulation that is a liquid, and showed at least as good of results as the initially early promising hydrogels that did also show good results. The early feasibility study results, as I have just shown you, are outstanding. This is an easy procedure to perform. It takes me between about 8 and 12 minutes to inject this into the disc. The breakthrough status was designated by the FDA, which really helps out in terms of the speed of the approval of this, and hopefully we'll start injecting this summer. As we've seen, hydrogel can provide fairly profound improvements in pain and function, and it can be used with any degree of disc degeneration. In fact, it provides good improvement in pain and function with any degree of disc degeneration. And unlike a lot of the products, is ideally suited for patients with severe disc degeneration, because this can act as a surrogate for disc tissue and fill the lost disc tissue up. Sometimes when you put patients prone, they have a vacuum disc. This is very good to provide a supplement to that lost disc tissue. And finally, on a personal note, there's not many technologies I think about probably every week for the past 15 years that finally reemerged, but this is the one and only that I know of, and I'm very happy for it. With that, we will take some questions, and I appreciate your attention. Thank you very much.